Welcome back, class. Mr. Rasmussen here. In this lesson, as you can see above me right up here, we're going to talk about this idea of meiosis. Yesterday, in the last lesson you looked at, we talked about this idea of mitosis and reproducing. Mitosis was really important to us for doing things like repairing lost skin cells. So if we get a scrape on our arm, we can repair those skin cells. It's also really good for us if we want to grow past the single-celled organism, which I hope most of us do. We want to become these complex multicellular organisms. Well, meiosis is important to us as well. Meiosis is important to human beings in the fact that we use it to reproduce. We use it to create egg cells and sperm cells so that when we reproduce, we don't have offspring that have lots and lots of chromosomes. So we use meiosis to create egg cells and sperm cells. And we use it in a different type of reproduction than mitosis. We use it in what's called sexual reproduction. And so where I want to start today, I just want to get an overview of asexual versus sexual reproduction. So if we talk about asexual reproduction is reproducing with only one parent. So we're not going to have two parents. We're just going to have one parent producing offspring, which is good, which has some benefits, and it has some negatives. Some examples of this would be a jellyfish. Jellyfish reproduce by what we call budding. And how they do this is this. If we have a parent adult jellyfish here, a new jellyfish is going to reproduce by just sort of budding off of it or creating a bud. So a new one is just going to start budding out of the old one, and eventually the new one's going to pop out and live on its own, and we'll have two jellyfish. So that's called budding. Now, don't get me wrong, jellyfish can also reproduce sexually, but sometimes they reproduce asexually during a part of their life cycle. Another example would be down here. These worms are called planaria, and they actually reproduce by what we call fracturing. So what happens is if we took a knife and we cut this planaria in half, both halves, both this half and this half, would form new organisms. They would both develop, even though this side has the head, a head would develop on this side, and they would create two brand new organisms. But there was only one parent here, so it only started from one parent. So asexual reproduction creates genetically identical offspring. Both the jellyfish that budded off and these planaria here, they're both the same. They have the exact same genes as the parent. Now this is good and this is bad for a couple reasons. Some benefits of this are it's really quick. If you want to reproduce a lot of offspring, you would do it by mitosis. It happens really quickly. You would do it by asexual reproduction. Bacteria can reproduce extremely fast. They can go from a handful of bacteria to millions and billions of bacteria within a matter of days, and that's how we get sick so fast. And the other thing that asexual reproduction is good for is you don't have to find a mate. And so organisms living in extreme environments or a lot of parasitic organisms that live inside of other organisms, they might reproduce asexually because they don't have to find a mate. There's no eHarmony of the liver or anything like that. So they would have to find a mate. They wouldn't have to find a mate in this case. Asexual reproduction is different than sexual reproduction. Sexual reproduction produces genetically different organisms. So your offspring are going to be different than the parents. And it does this by uniting sperm and egg together. So if we look over here, uniting sperm and egg, and each of these are going to carry half of the genetic information, half. So right here, sperm coming from dad is going to contain half the information or half the genes, and right here the egg is going to contain half the information or half the genes, and so therefore we're going to get two halves combining to get a whole, so therefore we have a little bit of genes from dad and a little bit of genes from mom each time we do sexual reproduction. The benefits of this are we get a different combination of genes. We get to mix up the genes each time. That's good because hopefully your offspring are going to be better suited to survive than you will. And when you have children in the future, you'll hope that they're better suited to live in this world than you were, or they'll hope that they'll be more successful than you are. So that's what sexual reproduction gives us. It gives us a recombination of the genes, a mixing up of the genes, and hope that those genes will actually be better. Okay, let's take a look at these meiosis stages here. In these meiosis stages, a couple things I want you to know. One, it's just like mitosis, except for it happens twice. There's two stages. There's meiosis 1. Oops, that one disappeared. There's meiosis 1 and there's meiosis 2. So you'll notice down here, you'll see things like early prophase 1 or prophase 2 over here. So they happen twice. So there's going to be prophase 1 and then prophase 2. But they happen in the same order. So after prophase comes metaphase. After metaphase comes anaphase. After anaphase comes telophase. So we're going to run through the same steps, except for when we get to the end, we're going to do it all over again. So after telophase 1, we're going to run into prophase 2. And then after prophase 2, we're going to go into metaphase 2. And after metaphase 2, we're going to go into anaphase 2. So, same thing. Easy to remember. The same things happen in the stages with a couple exceptions. One, here's the exception. Metaphase 1. If we look at this, prophase, same thing happens. Chromosomes condense down. The centrosomes move to the poles to prepare to divide. The spindle fibers appear. Over here in metaphase 1, the difference is on how they line up. And what's going to happen is each of these replicated chromosomes are going to move together. They're not going to split at the centromere yet. They're not going to break at that centromere. Over here in metaphase 2, over here, if I can find my pen again, over here in metaphase 2, that's when we actually break the centromeres. That's when we actually break the centromeres. So up here, if we start with 4 chromosomes in here, so if you can see here, we got 4 chromosomes. So we start with 4 chromosomes up here, and now we have 4 replicated chromosomes eventually. And in the first split, we then have 2 replicated chromosomes in each of these. There are 2 replicated chromosomes. And then we go through it again, and then when we start again in prophase 2, there's 2 replicated chromosomes. And now, at the end, we get 2 unreplicated chromosomes, or those centromeres split, and now we have 2 unreplicated chromosomes. So we start with 4, and we end up with 2. So we have divided the number of chromosomes in half. That's the important thing about meiosis. In order to create egg and sperm cells, which are eventually going to come together to form a zygote, we need to divide those chromosomes in half so we don't have zygotes with, in our case, 92 chromosomes. Another thing that happens that's kind of interesting is this idea of crossing over. Crossing over, and this only happens in meiosis. And what happens is we get 2 chromosomes right here that line up next to each other, 1 and 2, and they become intertwined. They become wrapped up in each other. And what they do is they swap parts of their chromosomes. So you can see here that they've traded parts of the chromosome. What this does for us is this further mixes up the genes. This really helps to mix things up and to make sure that the genes we pass on to our offspring are further jumbled up. Because remember, the benefits of meiosis are jumbling up, or the benefits of sexual reproduction are getting a genetically different offspring. Crossing over helps us further mix up some of those genes on those chromosomes. When we're done, it ends up looking like this. You can see how we have part of this blue chromosome attached to the red one over here, and part of this red chromosome attached to the blue one over here. So we have crossed over these 2 parts of the chromosomes and ended up with genetically different organisms, or a very different chromosomes. If we look over here, I just want to show you, to end here, I want to show you the results of meiosis. So we start with 4 chromosomes, and then we replicate them and cross them over. So now we have these crossed over chromosomes that are replicated. And then in the first set of divisions, we end up just keeping the replications together, or keeping those centromeres intact and the sister chromatids together. But now we have 2 chromosomes, or 2 cells that are replicated. And in the second set of divisions, we divide these up. Which chromosomes go into which cell are completely random. Completely random. So that when egg and sperm meet, it totally depends upon which egg meets which sperm, which combination of genes you're going to get. So which chromosomes go into which cell are random. Therefore, which organism is created from this sexual reproduction is random. That's meiosis, class. If you have any questions, feel free to ask me. Let me know. Thanks. Have a good day.